As a review, polymerase chain reaction is a process that creates many, many copies of DNA very quickly. So for instance, if you get DNA from a crime scene or from a person and you want to do genetic testing, you need to make many copies of the DNA you collected. And that process, or the process that does that, is PCR. So in polymerase chain reaction, you load your DNA into a machine called a thermal cycler, and that thermal cycler cycles through a series of 3 steps. Denaturation, which heats the DNA to break the hydrogen bonds and open it up. Annealing, where you cool your DNA and you use a primer that targets a specific gene. So let's say you want to test to see if somebody has Huntington's disease. You just want to target and copy the gene that contains the Huntington's gene. Okay, so you would find the gene you wanted to copy, make a primer made of DNA nucleotides that matched through complementary base pairing with that gene, and then that highlights the area to copy. So in annealing, the primers attach. Then in extension, you use a special DNA polymerase called Taq polymerase, which is from a thermophile bacteria they found in Yellowstone. And this polymerase is just like our DNA polymerase, but it can withstand high heats. And so that Taq polymerase attaches, and using nucleotides that are just found in the mixture you added, they make copies of the DNA. And then this process is repeated 40 times or more, and you very quickly can make millions and billions of copies of the DNA. Now, this video is really about what happens once you have all of these copies of the DNA. Once you have all these copies of the DNA, you want to see, let's say, was this person at the crime scene, or do they have Huntington's or something like that? So to visualize and see the DNA, one of your options, although there are many options these days, is to use a process called electrophoresis. Okay, so electrophoresis will be the main topic for this video. And electrophoresis is a process that separates molecules like DNA and proteins based on the size of the molecule and charge of the molecule. So we're going to specifically talk about DNA today. And as we know, this is a DNA nucleotide. And a DNA nucleotide is made up of a phosphate group, a deoxyribose sugar, and a nitrogen base, A, T, G, and C. But the most important thing to know about this nucleotide for the purposes of electrophoresis is that this phosphate is negatively charged. And the overall accumulation of all the phosphates in the DNA make the DNA molecule negatively charged as well. Proteins are a little trickier because you can have charged amino acids that are negative, positive, neutral, etc. So proteins don't usually have a uniform charge. If you want to do a protein electrophoresis, you usually have to do something to the protein to make it all positive or all negatively charged. But that's a conversation for a different day. Today, we're just talking about gel electrophoresis with DNA specifically. So we're going to talk about DNA nucleotides. Okay, so if you want to run a gel to see if somebody has a specific type of genetic disorder, if they were at the crime scene, if they're the parent, etc. First of all, you need many copies of your DNA, okay? And you don't want to have your entire genome. You want to have fragments of DNA. There are 2 main ways to get fragments of DNA. One is, like we talked about earlier, you can target a specific gene and just copy that gene using PCR. Another thing you can do is you can cut out specific sequences of DNA using something called restriction enzymes. So we'll start with one of our first DNA tools, which is a restriction enzyme. Restriction enzymes are naturally found in bacteria. And they act as their immune system by destroying foreign DNA. Right? So if this is your bacteria right here, this is the bacterial chromosome. and this bacteria is infected suddenly, okay? And it could be infected by a bacteriophage, right? We talked about this earlier. This is in the context of the Hershey and Chase experiment, this is a virus that infects bacteria called a phage. So if this phage inserted its viral DNA inside the bacteria, this virus is trying to infect this bacteria with its DNA, just like we might get infected with a virus, right? So what happens then is this bacteria needs to fight back. And one of the ways it fights back is by using restriction enzymes. So these little restriction enzymes come over, they attach to the viral DNA and they destroy it. So in that way, they act as a bacterial immune system. Okay, and each of these restriction enzymes find a specific palindromic sequence on the DNA um, to bind and break it apart. And when we talk about palindrome, it's the same thing forward and backwards. Like taco cat is the same forward and backwards, right? Or mom, or race car. These are all examples of palindromes. So when we think about common restriction enzymes that are used to cut up DNA, these are some of the common restriction enzymes. So these right here are the names of the restriction enzymes: EcoR1, BAMH1, N3, and PST1. And these over here are the restriction sites. And a restriction site is where the restriction enzyme cuts. And it's only a palindrome if you think about it as being a palindrome forwards and backwards. So for example, with EcoR1, the EcoR1 restriction enzyme finds this location on the DNA. Let me make it, double-stranded. So here's the double-stranded section it finds. And you can see it's a palindrome, GAATTC, because this side, GAATTC, is palindromic backwards, right? So it finds this location on the DNA, and it cuts like this. And it leaves these jagged, like, kind of single-stranded pieces of DNA plus a DNA on either side of it, right? And then, for example, BAMH1 finds this restriction site on the DNA. And we see how it's palindromic, and it cuts as well. So each of these restriction enzymes cuts the DNA at a specific site. And because of that, you get DNA of different sized pieces. So one way to get DNA of different sized pieces is use restriction enzymes to cut it up, then run it through a thermal cycler and make copies of those pieces. Another way is just to use specific primers in PCR to make those different sized pieces. And anyway, once you have those different sized pieces, you can run them through a gel. But I want to relate this to looking at genetic disorders or looking at other kind of differences in people's DNA. Okay, so in your DNA, you can have DNA that is used to code for a gene and DNA that is not used and does not code for a gene. Okay, so for instance, this could be a gene right here, okay, and this could be a gene for, I don't know, some sort of brain protein. So here is gene 1 for person 1, right here. But in the middle of your gene, you sometimes have repeated areas of DNA that don't mean anything. They're called random repeats, okay? So this person might have a bunch of random CAT repeats in the middle of their gene. So when this person's DNA, the sample 1 from person 1, is cut using restriction enzymes, okay, here, right here, might be a restriction site. So here's a restriction enzyme site. Here's another restriction enzyme site. When this person's DNA is cut with a restriction enzyme, you'll have a DNA fragment of this size, right? let's say person 2, here's gene 1 for person 2. They have the same DNA that matters, that codes for a specific trait, but this person has fewer repeats here. So they'll have the same restriction site on either side, and will use the same restriction enzyme to cut their DNA, but their DNA fragment will be smaller. Not because their gene that codes for the brain protein is any different, but because this person has random extra repeats in the middle of their gene and this person doesn't. Okay, so your genes could code for the same thing, but have different areas of repeated information. Okay, so basically, one of the things to note here is that repeated sequences, for in this instance, like CAT, okay, can vary between people. And this can make the DNA fragments for the same gene different lengths. And again, if you cut the DNA first, you would still have to do PCR to increase the DNA amount, right? But then you could load your fragments into a gel. A gel is kind of like Jello-y, right? So this would be a gel right here for electrophoresis. And this gel is it has like micro beads in it. And you can get more or less dense gels with bigger or smaller microbeads, depending on if you want to separate the DNA differently. Okay. And then you're going to load the DNA fragments into these wells. These are little holes in the gel. Right? And each of these count as a lane. The DNA is going to be loaded in here, and it's going to move in this direction, because what's going to happen is you're going to place an electrical current. You're going to run an electrical current through this gel. And the way you're going to do this is this gel is going to be suspended in a fluid called a buffer. And this buffer is a salt solution that conducts an electrical current. So let's see, how do I want to say that? The gel is placed in a salt solution, the buffer. And then we're gonna add an electrical current. Okay. And so what's going to happen is the electrical current is going to flow in such a way that this side of the gel is going to end up having a negative charge, and this side of the gel is going to end up having a positive charge. So you load your DNA in these wells, okay? And DNA is negatively charged, so the negatively charged DNA is going to move towards the positive end of the gel. But the genius of this is that the DNA will move at different relative speeds and velocities based on size. So each of these is a lane. Okay, the DNA starts here. This could be lane 1 here, and it moves in this direction. The next DNA starts here in lane 2, and it moves in this direction down the gel. Okay, so let's add that about the DNA first. The DNA is negatively charged and moves towards the positive end of the gel. Okay, so the DNA is moving in this direction, right? And then the fragments are going to be separated based on their size, which is going to impact the rate of movement. So what actually happens is that the bigger pieces of DNA don't move as quickly as the smaller pieces. Because remember, this DNA, like if this is your gel right here, and the gel is filled with little beads in it, okay? A bigger piece of, and it starts here, a bigger piece of DNA is going to have a hard time moving around these beads, whereas a smaller piece of DNA can wiggle through the beads more quickly. So bigger pieces of DNA are not going to move as fast as smaller pieces of DNA. So the distance traveled is related to the size of the DNA. So if we look at this lane right here, right, this is the smallest fragment because it traveled the farthest. So the smallest DNA travels the farthest. And this is a large DNA fragment because it travels less of a distance. So maybe like this piece of DNA is like 6,000 base pairs, and this is like 3,000 base pairs, and this is 1,000 base pairs, and this is 500 base pairs or something, right? So the smaller DNA travels farther than the larger DNA. Okay, larger fragment doesn't travel as far. The other thing to notice is that the width is not about the size of the DNA, it's about the amount of DNA. Right? So this is more DNA, and this is less DNA. Right? So for instance, this little skinny piece right here is bigger in base pairs than this piece, but there's less of it. Okay? So this could be like 10 pieces that have 800 base pairs in them, and this could be like one piece that has like 4,000 base pairs in it, right? So this has more nucleotides in it, but the DNA in that area is smaller, whereas this piece of DNA right here is bigger, but there's less of it. Okay. It is true that a lot of times you get a thick band in the bigger pieces because they just have a lot of base pairs clumped in that area. So then based on this gel electrophoresis, you can read this DNA and you can say, okay, if who's related, which people have the most DNA bands in common, right? Like these two individuals both had this DNA piece, but they're missing this DNA piece, right? If it's a crime scene, the DNA fragment from the crime scene the DNA fingerprint from the crime scene should match the DNA fingerprint of the suspect if you think that they were at the crime scene, right? So you can use this to do a couple things. You can use this to figure out paternity. You can use this for crime scene identification. You can use this for evolutionary connections. Are people more or less related to each other? You can also use this to say, hey, if you have this gene with a lot of inserts in it, you're going to develop Huntington's. If you have this gene with very few inserts in it, you're probably not going to develop Huntington's. So you can run these DNA pieces through the gel, and if people have DNA that goes farther, then their bands are probably indicating they don't have Huntington's. And if you have a DNA fragment that doesn't go as far, you're like, maybe you are going to develop Huntington's. So it's also a way to do kind of genetic disorder analysis, data analysis, and things like that. Thank you so much for listening to this video on gel electrophoresis. Have a great day.